My name is Dario Pisignano. I'm the Spoke leader of the Spoke 4, and I will just give you a brief introduction about the general aspects of this program, and then, of course, I keep most of the time for the technical presentations from our colleagues. Okay, so this is the content of the presentation. Just a few words about the general organization of this project. The coordination is at the University of Pisa. Then we have the major, basically the major Tuscany University, Florence, Siena, the Scuola Normale Superiore, two institutes of CNRR, the Nanoscience and the Applied Physics Institutes, that are based respectively in Pisa and in Florence, and the Italian Institute of Technology. The overall budget of this program is over 8 million euros, and it's organized, as you know, all the spokes are organized according to these milestone structures. Here, basically, we have three main sub-projects from the technical viewpoint. The first one is on intelligent nanoagents, so basically new carriers for drugs, for both diagnostics and therapy. The second big sub-project is on manufacturing, so technologies to manufacture devices for nanomedicine. And here you see that the focus is on the throughput of these technologies. So we want to have a high throughput for the methods. And then we have a third sub-project on so-called precision nanomedicine platform, which means methods to characterize all the products, so the nanotech products with multiple informations at different length scales and for customized applications. We also have, as all the other spokes, some cascade, we had already some cascade calls that close with 5 projects funded for an overall extra budget of about 2 million euros. What are the challenges? What are the motivations here? So first of all, the first task, as I told you before, intelligent nanoagents, carriers, and receptors for the several application fields. Here, the idea is to synthesize new products, nanotech products that can be effective tool from both the therapeutic and the diagnostic viewpoints, and possibly in the long term also improving the efficiency and safety of these of conventional drugs. We often say that the Spoke 4 set the most blue sky and long-term technological visions of the Tuscany Health Ecosystem, but always with a close look to specific application. The second big task is about, as I told you before, high throughput and advanced manufacturing technologies. These are especially bioprinting methods and both 3D and so-called 4D, so technologies to print materials and devices with adaptive behavior changing over time and responding to external stimuli. And then the characterization, the combination of different techniques to empower probes and biosensing new units. This is also the third part where we put the different ingredients together within bioreactors, especially on models for modeling the blood-brain barrier. And this is the kind of overall scientific thread, so developing of products of nanoparticles, not using animal models anymore, but using some sort of microfluidics, advanced microfluidics setup or bioreactives with intelligent scaffolds to culture cells, and setting everything together to finally propose a patient-specific treatment in the long term. So this is the broad thread, the broad scenario that keeps together all the different sub-programs of these Spoke. And transversal goals are important as well. So we decided to invest a lot of effort in commissioning and set up a new laboratory infrastructure. that is delocalized in the different partners, and this is of course open to access to all the ecosystems, including companies. And I will give you an example later, but we are talking about correlative microscopy, bioprinting, biophotonics, surface analysis, and so on. And this is the first goal that is almost accomplished now. So everything is running and interfaced in a closed network of competencies and instruments. The delivery, the implementation and the delivery of optimized synthetic procedure for intelligent nanoparticles, we use intelligent here as a way to say that they are capable to respond to stimuli. That can be optical, chemical. We don't want a static behavior, but something able to be activated in a as controlled as possible way. And with optimal loading properties. There are several groups, many groups actually in the program that are working on different classes of nanoparticles to have these addressed from different viewpoints and different chemical approaches. And then the fabrication part, as I told you before, so 3D printing in several ways, in several variants, and also 4-dimensional components. That means something that should be 3-dimensional in nature, in their spatial coordinates, but also able to evolve and to adapt to external environment and to bioreactor condition. What we had so far, this is actually not really updated because we are a little beyond this, but all the infrastructure of coordinated instruments is set and operating, as I told you before, and we had a lot of parallel approaches to deliver intelligent systems for drug delivery with different nanostructures involved that can be polymeric, lipid bales, plasmonic, and so on. Metal organic complexes and nanocellulose, so different kind of possible approaches to produce these kind of particles. We focus on two organ model, the eye and the lung, and we are engineering different devices in order to mimic the functional aspects of these two organ model, and to test drugs inside them. And we also started to integrate different components, which means placing nanoparticles in a framework given by bioreactors and integrated functional microfluidics, with also electrodes, possibly made with graphene and other two-dimensional materials in order to measure electrical characteristics inside the cell culture. So overall, we are quite happy because the Spoke is perfectly in line with its initial objectives, even in terms of expenditure, which is perfectly in line with the program. So everything is going quite well under this viewpoint, and I just give you a couple of examples of results. This is an infrastructural one. This is operating now at the physics department at the University of Pisa, and this is a correlative platform where basically you put together electron microscopy and confocal laser microscopy with a set of sample holders that are able to interface the two techniques and to provide you with a correlative information, so a multi-scale information, multi-method information, exactly in the same point of your sample. Here I show you a very quick example with the light-emitting polymer nanofibers, where you see an electron microscopy micrograph superimposed with a fluorescence information taken by the confocal part. And this is just a preliminary example of what you can do with this kind of equipment that can be for sure very interesting for a lot of biomedical characterization and cell culture, tissues, and so on. And another quick results is from a CNR group in nanosciences, in the Nanoscience Institute in Pisa. Here we focus on microfluidic components that can be done with several different 3D printing technologies. So this is important for us because this is fast in operation, and you can engineering, of course, as typical in additive manufacturing, you can engineering the different components of your microfluidic chip in a very quick way and doing a lot of prototypes in a fast approach. And let me see if there should be a video here. It's moving. You see, this is just an exemplary equipment, microfluidic equipment to produce emulsion that you can do with different chemical behavior. And you can also, you can also characterize this droplet distribution as test for drugs and small bioreactors in terms of the size distribution of the droplets and also the overall architecture of the emulsion coming out. So I would like to thank all the colleagues working on this other task, and you will see a lot of other results in the next talks that will be from Anna Piras from the Pharmacy University. From the pharmacy department at the University of Pisa. She will talk about some examples of the nanovectors we are developing for lung and eye delivery. Then colleagues Maria Cristina Ceccarelli from the Italian Institute of Technology in Pontedera. She will talk about monitoring 3D blood-brain barrier configurations, in particularly the maturation integrity in real time with the sensorized microfluidic device. And then Francesco Baldini from the Institute of Applied Physics of CNR will talk about smart nanostructures for intracellular thresholds, theranostics, and optical biosensing. And we will also have an entire session in the main conference that is named the Next Health Journey conference in the other room about tools and models for the development of therapeutic session where a lot of nanotechnology products are involved and the results from our spoke, and this will start at 5 p.m. in the other room, in the main room. Just a very quick perspective for the next 12 months. We have a lot of nanosystems and nanoparticles, as I told you before, so we need to characterize them in depth to assess not only loading efficiency, but also delivery and targeting efficiency, the validation of the chips in all their different functions, both in terms of fluid control and in terms of drug testing. And further development of all the big platforms that we develop for advanced precision characterization that as you can see here, involves a lot of different technologies to be placed as in parallel as possible to get multimodal information from the samples. And the final very important objective for us in the next months is improving our interface with companies that can be interesting in the technology transfer aspects and in scientific collaborations with the Spoke groups. So this occasion is really precious for us. So I come to the final acknowledgement. Of course, I would like to thank all the scientists in charge of this effort, and particularly all the members of the coordination group of the Spoke, Professor Baldini, CNR, Calderoni and Vozzi, the University of Pisa, Professor Cantini from the University of Florence, Professor Cardarelli from the Scuola Normale Superiore, and Professor Ciofani from the Italian Institute of Technology, and Professor Magnani from the University of Siena. And of course, all the PhD students and researchers that we are on the Spoke. We hired more than 20 young scientists, and they are really doing a big, an excellent work in the labs, and so they are really the most important ingredients of all this stuff. And all the organization of this event that is going great, and particularly Francesco Mazzini for the invitation and all the effort. So thank you very much, and I leave the stage to Professor Piras from the University of Pisa for the next talk. Thank you.